Well, good morning again. We are continuing, and now Professor Yang Wanping will give us, yes, the talk, Improving and Culturing Kelp for Bioethanol Production.
Welcome back again. So I'm going to talking about the real biology, not the geography. So this one is the kelp, or laminaria in Latin language. So we call it, I think 100 years ago or 50 years ago, we farmed the laminaria kelp with a method. It was called floating raft, floating frame, something like that. This kind of method has evolved into the floating net. This is the head rope. The buoyancy there to float the head rope. At this end, at both ends, it was fixed to the bottom of the sea. And between the head ropes is the pending rope or the hanging rope. No. This is the hanging rope. The kelp was inserted in between the skins of the hanging rope upside down. So the kelp there just grows from the top to the bottom. Is different from nature. So I'm talking about today, is breeding the kelp. Of course, before for eating, before for the production of alginate or the mannitol. Right now, it's for the bioethanol. Of course, it's about the energy. So how to do that? This solar energy is clear. And after that, we cannot change the solar energy into biomass. We have used some organisms. And then to the sugar. Sugar is polysugar. So with the help of microorganisms, it can be changed to the single sugar. And then with the help of microorganisms, it can be changed into ethanol, or called bioethanol. Of course, you can change it into oil, change it into coal, but it's a long time. Also, you can change it into the medicines. It's historically or just can do it right now. So we will focus on the bioethanol. So if we try to ferment or produce bioethanol, many biostocks we can choose. First one is crop starch, like the starch, like the sugar cane starch or big beet sugar starch, something like that. Sugar here we can ferment it into carbon dioxide, heat, ethanol, solid residues. Of course, we can process it into finally the ethanols. Before we can drink it, liquor, like anything, you can drink it. So this is the traditional ways. However, we cannot use it for producing bioethanol. Why? Because the starch is there, we can eat it. Maybe in this country you can use it. Maybe in the United States you can use the starch to produce ethanol for the benzene, for driving the car. However, in China, in South Asia, in the Asia countries, this kind of avenues is not sustainable. So next time, the other choice is how to say that we can make the ethanol from the wood. For chemicals components of the wood is lignin, cellulose. This is the three major components of it is lignin, hemicellulose, and cellulose. We can use for more than 100 years. Many scientists try to degrade or change the cellulose or cellulose into finally the glucose, the single sugar. So we can use the a cell factory. It was called the yeast. These Latin names. It was widely used in the industries. You see the PPP pathway. We do not want to know the details. PPP pathways and go into the glycolysis, separate the sugar, and then to the ethanols. The yeast can do this. The blue pathways yeast can do it. However, the yeast cannot use the cellulose directly. So the cellulose should be pre-treated, use the acid or the alkaline, something like that, into the hemicellulose, and then cellulose. It's not a single, it's a bio something like that, I cannot remember. Also, the cellulose can be pre-treated into the cell bios. Why? Also, it can be pre-treated into the glucose. Why not glucose? Because the yeast can use the glucose first. When the glucose was used, the ethanols was many ethanols was synthesized. In this case, the cell cannot use the cellulose. So in that case, we just use the techniques to change the cellulose into cellulobiols, two sugars, sometimes like, also cellulose. Then the cellulose can into the cell. We can use the transporter here into the cell, and then finally use the other enzyme. This was called the glucosidase, sometimes like, finally into the glucose, then goes to the ethanol. However, the yeast cannot use them. So we can introduce the genes from the other species, like this is also a yeast. It has the XR, XDH, and the XKS, 3 proteins. I cannot the full name of this kind of enzymes. Anyway, we can transform the yeast with the genes encoding these 3 enzymes. Also, we can transform the yeast with the 2 genes. It was from the other, this Neurospira, is a fungi, something like that. So we introduce the genes into the chromosome, we pre-treated the cellulose into this one, and then this is the whole system. In this way, the yeast can produce ethanol. This way, the method, the techniques is available, is there. I think not developed by us, but it's available currently. However, this pathway is still not sustainable. Why? 3 concerns here. One is pre-treatment. We need to spend money. Second concern is that environmentally will cause a lot of pollutions. And thirdly, tree is on the hills. If all the trees was used to produce ethanol, so it will cause a lot of problem in the ecological functions. So it's not sustainable. So third choice, we just think about the kelp, or the labnera, or the Chinese haidai, or the kombu in Japanese. Is carbohydrates, is polysugar, is brown algae or macroalgae. So if we use it as the biomass to produce ethanol, it has 3 advantages. One is we do not need land. The other one we do not need fertilizers. The third one, no fresh water was needed. So this is the big advantages. You have 2. If you try to use these advantages, the big if here, all kinds of carbohydrates of kelp can be fermented into ethanol. The components here is arginate, mannitol, and glucan. These 2 can be used by yeast, can be changed into ethanol. Arginate cannot. Also, the major components here is arginate, is 70% of the biomass. So how to do that? This is the structure, just illustration. You see, arginate here, mannitol here, and glucan here is polysugar. This is toll here is ethanol, something like that. This is also polysugar. Totally 70% of the dry weight. These are the percentages. Before the bacteria can change these 2 into ethanol. However, what you need to do is just the grinding and the mining into the liquid materials and then ferment it into ethanols. Before, the arginate cannot use it. So we have to try to develop one method to ferment all these kind of 3 major components into the ethanol. So how to do that? We have to get help from nature. So in nature, we have a species of bacterial species. It was called spingle molars. This is a single cell, something like that. I cannot remember, cannot understand what the Latin name means. On the cell of this kind of bacteria, it has a pit or a hole there. The arginate can go into this cell, goes through this channel into the cells. During this process, many proteins, some are just outside of the cell, some are inside the cell, working concertedly in cooperation, change this arginate into the monosugar. 2 of them here, one was called M, the other one was called G, something like that, So if this polysugar was changed into the single sugar, the cell, the bacteria can change it into the carbon dioxide, also produce energy, and then for the growth of bacteria. So we need to change it. How to change it? You see, here we can see clearly here. This is the arginate with the help of a group of proteins. The arginate goes into the cell, change into these 3 chemicals. I cannot. The full name is here. And then this is how to say. This pathway was called Nettler, Dodorov. These 2 scientists. This is a pathway. We know that the pyruvate is how to say the central molecules inside the cell. If we give the bacteria oxygen, it can be changed into acetyl CoA, and then TAC or TCA. This is the energy or the NADP or something else. It was called the cellular currency for growth. And some of them will change into the lactase. This is byproducts we do not want it. Anyway, it can do it, but where is ethanol? No ethanol there. So we still want to. The first thing we should do that is close this way. So no lactase was produced. And the second change is we introduced two genes, ADH and PDH3. These two enzymes, two proteins. We get the genes from this zymo-molars. It's single cell. Zymo means movement, something like that, So we introduce these two genes into the bacterial cells. So how to introduce the genes into the cells? We just construct a plasmid. In this plasmid, we have two genes here, promoter and terminator. It will express into transcribe into the mRNA and then finally into the enzyme. Also, we change the copy number. One copy there and 8 copies here. So these two enzymes will change the percentages so that they can, at the highest efficiency, change the pyruvate into the ethanol. Of course, you shouldn't give it the oxygen. So also the method is there. Why we not use it? Because it's a non-industrial bacteria. That means we can the fermentation efficiency is very low. So we cannot change it more. So in this case, of course, we try to change it into the other bacteria. It's E. coli. Everyone here maybe know the E. coli. It's a very important one. Also, it's in our body working with us, help us to survive on the earth. So how to change it? The background is here. For the E. coli, the glucan is sugar, polysugar, or the mannitol is ethanol, something like that. These two components can be changed into this kind of stuff. This is available in the cell. So we try to use the E. coli to produce ethanol. Of course, we close it first. How to close it? Just close the one. How to close it? Break down the genes on the chromosome. And the second one, we introduce these two enzymes. I just told you what it is. And then from pyruvate can be changed into ethanol. And the third very, very important thing is to, let the E. coli can use arginate. How to do that? We get to is, a hybrid protein. One is the arginate degrader, is an enzyme. Second one is a protein of the bacteria of E. coli. It was synthesized first inside the cell and then goes to the surface of the cell, serve as an antigen. So we make these two proteins come together. We just introduce the genes. We introduce genes somewhere here, and the proteins goes to here, change the arginate into oligoarginate, and then let it goes into the cells. Of course, E. coli cannot do it. How to do it? How to let the E. coli to do this? We just introduce the genes. This is not a single gene; it's a group of genes. In molecular biology, it was called cluster or just a group, operon, something like that. So this kind of gene is from where? From vibrosis. This bacteria was synthesized, was sequenced in the Shandong University, China. So we introduce this group of genes into the chromosome, then synthesize a group of proteins. Then E. coli can use the arginate, change the arginate into this one, then goes to this pathway, then to the ethanol. So of course this work is not done by us. The techniques and the method is available currently. So next question, where is the biomass? We cannot use the starch, we cannot use the wood. We try to use the kelp. So the kelp is here. Next question is it was from the harvest the biomass from the nature? Maybe here is okay, but in China, you cannot do it. Also, from a farmer, of course, in China, the only choice is from a farmer, from the kelp culture. So we can, in China, we have 3 places. Here is Liaoning and Shandong, our hometown, also our university located at. And also in the one province in the south part of China, these 3 places producing farmer kelps. The total area is these hectares is about 400 kilometers. It's just about the size of Jiaozhou Bay. You see here is a bay here. It's the size of the Jiaozhou Bay, about 400 kilometers. Just remember these numbers. So for us, how to do that? Traditionally we use the selective breeding, this method, to improve the kelp. The yield is 20 tons per hectare per year. It's about 35 of the theory yield is about 60 tons per hectare per year. That means if we change the yield from 20 tons per year per hectare to 59 tons per year per hectare, this is a window. This is how to say the range for us to improve or the breeding the kelps. How to do that? This is a big story in China. This is this guy, and of course this is how to say big scientist, of course. This is a rice. Traditionally, rice just about maybe like 500 kilograms each. How to say? It's a calculation. Anyway, that means the hybrid rice. Through this way, the yield of rice changed from how to say the base to fourfold increase of the yield. This kind of story should be can be adopted for the genetic imprint of the kelp. We call it hybrid kelp. Also, for human beings, like the marriage, different countries, different tribes, their kids should taller or stronger or pretty, something So this is a good word anyway in the genetics. So we can use it for the kelp. How to do that? This is a life history or life cycle of kelp. We can divide it into two parts. This part is how to say young sporophyte. That's what we into the major the old sporophyte. This we harvested. Also the other part, it was called the gametophyte. The root spores, male and female gametophytes, eggs and sperms, and then finally zygote or zygotes, and then change each year. Very importantly, we should remember here is this kind of gametophytes can be artificially cloned into the into the clones. This is what we did into we can separate the clones and then preserve it in the low energy, low orientations and at the low temperatures. So also this is what we did. You see, we get the gametophytes, cultivate it into the clones, and then preserve it or cut it into just amplify the biomass. Finally, we can spray it on the cement pole or just on the tree. We can make the young sporophytes. We can cultivate in the sea. This is one story. We can make the hybridization, get the hybrids. And the second one, traditionally, we get the sporophytes wash, and then just briefly air dried briefly, and then back to the seawater, and then move out the sporophytes, leaving root spores there. We put the cottons into the cement poles and get the young sporophytes here, and then cultivate and harvest the sporophytes. So along this way, the techniques is there. What we have did is that we get the clones and make the hybridization, get the hybrids, and get the seedlings from hybrids, and get the hybrid here. So in this way, we just get each year we make the hybrids and then farm it. We select out the best one. We breed three varieties. It was called Dongfang number 2 and number 3. The other one, it was done in 2007 and 2008. This is my students here. So we find that in this way, each year we have to make the hybrids. However, in the traditional systems, the farmers, this kind of techniques cannot be used directly to the traditional system because it's a little bit complex. So we changed our strategy. We go further. We get the hybrids and the self-crossing, selection and rising and farming, just a few rounds, like 3 rounds here. We get the best lines. This one just is not a hybrid. It was selected out of the hybrids. It's a purified lines. So usually we get the other 2 varieties. One is called the 90-1. The other one is number 6. Also, for this one, still have some these shortages. Why? Because it was from a different species. One is different species. If we try to use this kind of varieties to produce seedlings, the efficiency is very low. That means the hybrid can get the kids is not comparable. For example, the humans, different tribes, different countries come together. Their kids is stronger, but the the kids of the kids may become very, changing a little bit. So usually we change our strategies again. If we get the baseline, we just use it for 3 years. Well, that's it gone. Change it, already changed it, that's it gone. We separate the gametophyte clones from the baselines. We keep the gametophytes, and after 3 years, we reproduce this kind of varieties, and then farm it. So in this way, I think we have changed the breeding and the culture of the kelp in China. So we're back to the yields. Traditionally, it's 20. What we have done is 30. We use the hybrids. So 30 tons per hectare per year is almost 50 of the theory yield of the kelp. So we do one more thing we try to do is that for the kelp culture, the kelp is not living in the warm water. It inhabits, it grows well in the cold water in the winter times. So from here to here we culture it. Here is summertime, 3 months. So when the night is stronger, temperature is high. However, we cannot use it. So we try to breeding. Right now we try to breed in the other alga species or the other microalga species. It was high temperature tolerant. So in this case, we can double or culture 2 times or year-round culture. In this way, if the yield was doubled, it will close to 100% of the theory. It's about 60 tons per year per hectare. So if we just get a halfway. If we can touch the end of this kind of trial, we can get this kind of yield. So if this is the areas at present we farm the kelp. This is the million liters of the petroleum. It's about 236, 1 in 236. It's about 0.5% of annual petroleum consumption of China. If we use all the kelps to produce ethanol, we can change 0.5 of the annual petroleum consumption of China. So that's what we are trying to do at present. If we try to replace the petroleum to 5%, it's about 2,000 kilometers. Of course, it should be 2 rounds each year. This kind of area is just about 1 in 1,500 total sea areas outside governed by China. Totally is about 3 million kilometers. So I think it's a promise avenues to produce bioethanol to replace the petroleum in China at least. So what we have right now is about 1% of the total area of China. We try to increase the total acreage for culture, for farming the kelp. How to do that? The one way is culture it with the animals. It was called health animal, like some oil like this. This was abalone, oyster, clams everywhere, or some kind of fish. Of course, they produce waste. If we culture the kelp there, well, it will help the animal, also help to improve the quality of the environment. Here is somewhere in construction. In this area, of course, East Coast is seawater. Many pollutions there. Even the kelp can be cultured there. The kelp cannot be eaten. However, we can harvest the kelp for the production of ethanol. Also, this kind of the clean energy. Here is some big piles there, so we can fix the roofs between them and the culture farm the kelp in the open sea. In this way, we can increase the total acreages of for farming the kelps. If we can reach to 5% of the total areas of China, so we can replace at least 5% of petroleum consumption of China at present. So before we breeding kelp for food, for feed, and right now, 20 years ago for alginate manure production, right now for bioethanols. So any omics for kelp, omics because you can do something group by group, that means omics. MI means come together, or just one characters there. For kelp, any omics work, any work in omics we are doing right now, we have some of them. We get the high density sensitive map. This is one thing we have done. We have these varieties, the other varieties. Of course, they are genetically very distant. So we get we cross them. We get isolated gametophytes and get a mapping panel or just isolation populations. And then we use the technique, it was called IAD. IAD here. Of course, this is a second generation of sequencing techniques. I do not want to go details about the technique. However, we get 1 through n. It's about 200 individuals. We get the positions 1 through n. This n may be 10,000. We get, SMP1 through SMP maybe 10,000 or something like that. We use the computer to calculate the linkages between them. We get one linkage map, each about 5,000 SMPs, each about 99 microsatellites, and each site, because just short, the range is very short. So we exchanged each site to about 500 BPs so that we can develop chips or somewhere high efficient genotyping methods. So this is what we have done. Also, we try to sequence the genome because we try to do more. Unfortunately, we are later than the others. This one has been published by the other guy in the other institute. Our work is just ongoing. Well, this is some kind of a competition. However, we still want to go further to get the genome sequence and do more, to write more stories. Well, I'm going to the other. What kind of thing I'm trying to do? We can compare the life cycles of here. This Cladiomonas, this is a microalga. This is the volvox, also is a microalga, but it's multiple cells. Here is fence, everyone knows this, has a similar life cycles. This is mosses, similar life cycles. Here is this one. This is the kelp life cycle of kelp. So we can we try to get the genetic basis of the gametophyte sex, female and male. What's the genetic reasons to determine this kind of phenotypes? How to do that? We learn from the other species. Here is a yeast. Here is the is monoecious, means just a single sex, something like that. This is papaya. This is a plant. Asparagus, a plant we eat in the this is a plants. Mosses also is a plant, something like that. This was done before today. So they get they find genes or chromosome regions, or just a locus to determine the sex of this kind of species. Go deeper. For the Cladiomonas, for the volvox, for the leafwort, also a plant. They know this is the site to determine the sex. We can go to the genes or the genetic with the genetic loss, linkage the genes with the sex. So this is what was done for the other species. For the kelp, for the sex of kelp. So in this way we can understand that it should be this kind of region have different structures. Should have some genes differentially expressed between sex. Of course, there are sex associating genes. How to find this kind of genes? Well, this is my just designed. You see, we can get the separation already. It's there. It's for the SMP map construction. We group them into 2 groups. This one is male. This one is female. Each group 20 gametophytes there. We use the bulkhead segregants RNA seq, bulkhead segregants DNA seq. Is we get the different genes, the different markers and the different genes, and then we tightly link the genes with the. We get the tightly linked genes, tightly linked markers. We go back to the genome sequence. We go to the sequence scaffold. It's just a frame, something like that, Then we can find the structure, and then we find the expression patterns, the difference between genders, and then we can finally understand the sex determination mechanism of the kelp. Well, our ongoing work also include, you see. This is one species. The other species get the gametophytes, make the hybridization between them, get the sporophytes, and then get the self-crossing and the selection, get these 2 varieties. And the recombinant self-crossing, what I'm trying to do. Well, yeah, we get this. This is a population. We just get the recombination self-crossing lines. It's really like what we have done in the rice. Then we can find the chromosome fragment or the corresponding traits. Use the genetic loss, we get the genetic basis of economic traits. This is what we are doing right now. So, and then we clone the control genes, and then we use like a transformation to verify the genes or lock down the genes to find their functions, and then use a lot of methods there. So in this case, we try to find the genes to determine the traits of the kelp. Of course, they are the important traits associated with the economic values. So this is what we are doing right now. So for the omegas, some suggestion for international cooperation. To my point, my understanding, we have two. One is environmental organism diversity. Just the two professors at this beginning of this meeting, they find the relationship with the dust and the combination of plankton. So what's the composition of plankton? We can use this kind of genes, use the second generation segregation methods to linkate the chemical effects with the biological effects, and then we can go deeper. That's the one. Second one is ecological phenomena. What's their mechanism? We can get the DNA from the environment. We can sequence the DNA, either what called metagenome or bigger genome or changing genome, something like that, you know. So we can get the genes there, annotate the genes, know their functions, and then those are the pathways. Then we can predict the pathways. Then we can know what stories happened in the nature. Of course, the third method is the single cell based techniques. I can understand that, so I wouldn't to say any words about that. Well, that's what I'm trying to talking about. Okay. Into hemicellulose and then cellulose. It's not a single. It's a or something like that. I cannot remember. Also, the cellulose can be penetrated into the cell bios. Why? Also, it can be penetrated into the glucose. Why not glucose? Because the yeast can use the glucose first. When the glucose would use it, the ethanol was many ethanol was synthesized. In this case, the cell cannot use the cellulose. So in this case, we just use the techniques to change the cellulose into cellulobios, two sugars, sometimes like that, you know. Also cellulose. Then the cellulose can into the cell. We can use the transporter here into the cell, and then finally use the other enzyme. This was called the glucosidase, something like that, you know. Finally into the glucose, then goes to the ethanol. However, the yeast cannot use them. So we can introduce the genes from the other species, like this is also a yeast. It has the XR, XDH, and this kind of avenues is not sustainable. So next time, the other choice is how to say that we can make the ethanol from the wood. You know, for chemicals components of the wood is lignin, cellulose. This is the three major components of it is lignin, hemicellulose, and cellulose. We can use for 100 years, many scientists try to degrade or change the cellulose or cellulose into finally the glucose, the single sugar. So we can use the a cell factory. It was called the yeast. These are not names. It was widely used in the industries. You see the PPP pathway. We do not want to know the details. PPP pathways and go into the glycolysis, separate the sugar, and then to the ethanol. The yeast can do this. The blue pathways yeast can do it. However, the yeast cannot use the cellulose directly. So the cellulose should be pretreated. Use the acid or the alkaline, something like that, you know, into to the bottom of the sea. And between the head ropes is the penny rope or the honey rope. No. This is the honey rope. The kelp was inserted in between the skins of the honey rope upside down. So the kelp there just grows from the top to the bottom. Is different from nature. So I'm talking about today, you know, is breeding the kelp. Of course, before for eating, before for the production of arginate or the mannitol. Right now, it's for the bioethanol. Of course, it's about the energy. So how to do that? You know, this solar energy is clear, and after that we cannot change the solar energy into biomass. This we have used some organisms, and then to the sugar. Sugar is polysugar, so with the help of microorganisms, it can be changed to the single sugar, and then with the help of microorganisms, it can be changed into ethanol, or called ethanol. Of course, you can change it into oil. change it into coal, but it's it's a long time. Also, you can change it into the medicines. It's historically or just can do it right now. So we will focus on the bioethanol. So if we try to ferment or produce bioethanol, many biostocks we can choose. First one is crop starch, like the starch, like the sugar cane starch or big beet sugar starch, something like that. Sugar here we can ferment it into carbon dioxide, heat, ethanol, solid residues. Of course, we can process it into finally the ethanols before we can drink it, liquor, like anything you can drink it. So this is the traditional ways. However, we cannot use it for producing bioethanol. Why? Because the starch is there, we can eat it. Maybe in this country you can use it. Maybe in the United States you can use the starch to produce ethanol for the benzene, for driving the car. However, in China, in South Asia, in the Asia countries,
well, good morning again. We are continuing, and now Professor Yang Wanping will give us the talk, improving culture in kelp for bioethanol production.
Welcome back again. So I'm going to talking about the real biology, not the geography. So this one is the kelp, or laminaria in Latin language. So we call it 100 years ago or 50 years ago, we farmed the laminaria kelp with a method. It was called floating raft, floating frame, something like that. This kind of method has evolved into the floating net. This is the head rope. The buoyancy there to float the head rope. At this end, at both ends, it was fixed